Well, hello everyone, and thank you very much for asking me to give this talk. I'm really sorry I can't be with you in person. Now, I have heard people say that male ferns are boring, and maybe they're not as pretty as some more lacy ferns, but I hope to persuade you that the scaly male ferns are a fascinating group and well worth further study. So my interest in them started about 15 years ago when I began to learn about them from Ken Truran. And just before Ken died in 2010, he asked me to see that his guide to the complex was published. And I co-edited that with Anthony Pigott, and it was published in 2014. Well, since then, I've done quite extensive field work on this group with Tony Church and with Roger Golding, who some of you might know. Last year, I completed a research master's on the diploid taxa, but because of travel restrictions, I wasn't able to include any Irish specimens then. Well, I'm now registered for a PhD, and I've been able to spend 2 very intensive weeks of fieldwork travelling around Ireland. But there are still many places that I haven't visited, so I have drawn on the BSBI distribution maps to fill in the gaps for this talk. Move on. Why won't it move on? Sorry, Jim, it doesn't seem to want to move. Ah, there we are. Good. Paul gave me a list of what he wanted me to talk about. So this is the plan. And because the taxonomic history is very long and involved, this is going to be a very brief summary. In the middle of the 1800s, people thought that there was only one male fern, but a man called George Wollaston, who studied them in detail, proposed there were 3, and he used different names, but basically it was our common male fern, the mountain male fern, and the scaly male fern. His theory was proved correct by this lady, an amazing woman, Irene Manton. She was professor of botany at Leeds University. So she showed that he was right. There was the common male fern, the mountain male fern, but that the scaly male ferns actually consisted of a group. She was an expert cytologist, and much of what we know about the genomic composition of the scaly male ferns is still based on her work. And she also discovered apomixis, that's reproduction without sex. So where the spores have a full complement of chromosomes and the new sporophyte grows directly from the gametophyte without fertilization. So the parent clones itself. When she discovered that a German, Herr Dopp, had already described this, she was a very fair-minded person. It became known as the Dopp-Manton premeiotic endomitosis. So she knew the scaly male ferns as Dryopteris borreri, but in 1979, Fraser-Jenkins published a name change to Dryopteris affinis because he discovered that the type specimen was collected in Madeira in the 1830s and was named Nephrodeum affine. And that means that the affinis predates Newman's borreri. Now, you may be familiar with Chris Page's 1997 taxonomic treatment. He based his work on that of Fraser-Jenkins. So now just let's look how we recognize affinis from those other male ferns. most scaly male ferns have this dark spot where the costa, that's the midrib of the pinna, joins onto the rachis. It's not absolutely always true, but it's a very good rule of thumb if you see that dark spot. Our common male fern, on the other hand, is usually much less robust and it doesn't have the dark spot, although the whole of that rachis can sometimes colour brown later in the year. The other thing is that the indusia of the common male fern are fairly flimsy and drop off fairly soon. They often have rather spreading edges. Then the mountain male fern, I think it's very rare in Ireland. It doesn't have a dark spot. The indusia are tucked under and they have glands on the edges. The indusia are normally limited to the inner half of the pinnule. So I was quite excited in Waterford because I thought I'd found one, but I've checked it out and I'm afraid it was a common male fern and not a mountain male fern. But I'd be interested to know if anybody has seen it in Ireland. So to carry on with the taxonomy, it is difficult to know the ploidy level, so whether it's diploid, triploid or higher, just by looking at a plant. And so Clive Jermy and Anthony Pigott, along with Jermy and Camus, they proposed this term morphotypes. Now, this is not a legitimate botanical term, and it thoroughly annoyed Christopher Fraser-Jenkins. Interestingly, convexa, this form which looks like it's stamped out of plastic, is quite common in the north of England, but I don't think I've seen it in Ireland. So if anyone has seen it, I'd be really interested to know. And there's more about Arranensis later. Now, two Americans, Battle and Buck, argued that if taxa had different genomic compositions, they should be classed as separate species. And so Fraser-Jenkins was eventually persuaded to publish the subspecies at species level, each with their own subspecies. So this is the basis of the taxonomic treatment in Stace 3rd edition. And these taxa can be recorded on the BSBI database. Now, the guide that Ken Truran had prepared actually describes 11 distinct taxa and 7 hybrids. This guide's out of print inside a year, but it is available as a PDF on the BPS website. And this is Ken Truran next to the first recognised plant of Dryopteris pseudo-disjuncta in the UK. It's been recognised as a species in mainland Europe for some time. Christopher Fraser-Jenkins thinks he found this at Drumber Bridge, but we've searched for pseudo-disjuncta and we haven't been able to find it. If anybody thinks they have, again, would be really interested. So moving on, And this slide is in the handout, so no need to copy down all the names. Peter Sell published all the previous subspecies and other species that have been described. He published them as species. The numbers here refer to the ploidy level, that is the number of sets of chromosomes each of those has. So that was 2018, but then in 2019, Stace 4th edition really followed Fraser-Jenkins and made those species back to subspecies. And quite a few of us don't think that this is very helpful. It does really upset the naming of the hybrids, because hybrids with the old subspecies have now just been sort of sunk. Anyway, for this talk, I've chosen to adopt the nomenclature as in Sell and Murrell. Now, this rather complicated slide is also in the handout, and I hope it'll help you to understand the relationships in the affinis group. So those sexual species, which are not scaly, are shown in green at the top. Dryopteris caucasica is not known at present in Britain or Ireland. So either it's become extinct, or the filix-mas was formed nearer to the Caucasus and has spread. If it invades your garden like it invades mine, I think you agree that it is quite a spreader. So here, the three main species of Scaly Male fern are in red, and each genome is represented by a letter on the diagram. Now, each Scaly Male fern has a genome from this unknown affinis ancestor, which could be Dryopteris williciana or something related to it. So I've said that these species are apomictic, but what happens is that the gametophyte can actually produce male cells that swim off and can fertilize the sexual species. And this is how the tetraploid and pentaploid hybrids arise. So the hybrid between Dryopteris affinis and filix-mas, the cross complexa, is really fairly common, and it's fairly widely recorded in Ireland. But so far, we haven't found either of those pentaploid hybrids in Ireland. And if you think you've found these, then please let me know. So those hybrids have a really high proportion of abortive spores, and that is, if you think you've got a hybrid, then you really need to look at them when the spores should be ripe and see if it's got good spores or if they're all shriveled and abortive. However, hybrids do produce some viable spores, and Libor Eckert in the Czech Republic has found that Cross critica actually can produce spores with 5 sets of chromosomes and with 2.5 sets of chromosomes. So it is possible that new forms are arising from these hybrids all the time. So just to summarize the problems, So I was very unhappy with the taxonomic model, really. I don't think it's helpful for recorders, and hence my current study. So this is my title. And my research questions. So this is primarily a morphological study. I'm going to deal with my methods for the first 3 questions. I'm afraid going into the DNA is just a bit too much for the time available. Before you can study anything, I think you need to have defined it well, and unfortunately neither Cell nor Stace seem to have provided any morphometric evidence for their taxonomic treatments. So the first thing when I collect a frond, I obviously record where it is, its habitat, the number of fronds, number of crowns, and then take a frond and measure it. And here's a summary of the measurements. And also I measure the degree of adnateness of this first lowest basiscopic pinule. And something that I'm not sure has been looked at before, but Roger Golding suggested it, the number of vein endings actually in the auricle, that's the little lobe at the base of the pinule next to the rachis. And that does seem to separate out the taxa. I've also coded toothing, crimping, and the appearance of the indusia, that's the cover over the spore cases after the spores have been dehisced. What did surprise me in my study of the diploids was that there were many significant differences between the taxa in the morphometrics, and that wasn't including borreri or Arranensis. So more measurements. I've been measuring length and width of the stipe scales and the length of the rachis scales. When I wondered how to do it, it was Roger again who suggested using a glue stick on paper and then sticking the scales onto it. And so I got my Pritt stick out of the drawer and stuck the scales down. And as soon as my plastic ruler went anywhere near, all the scales jumped off the page onto the ruler. So that didn't help very much. But a new Pritt stick sorted it all out. Now, once I've done the measurements, I've been creating a virtual herbarium by scanning the fronds in sections. And I've included this one because it's a nice small frond. It just went into 2 sections. It's scanned top and bottom. Some of the fronds are so big, they go into 5 sections, and that's quite a lot of scans. But it has been enormously useful to go back to compare fronds later on. In addition to that, I take some digital microscope images So I can see the stipe and the scales. I actually do close-ups of the lowest pinnules, which I haven't got on this slide. This is the same frond as that Arranensis from Tahila. I think you can see that the pinnules are really quite crowded. Now then, knowing the ploidy level really helps with identification. And in 2015, Arnaud Bizot published these results, and he'd measured hundreds and in some cases thousands of stomata to come up with these averages. This is something that with a good quality microscope you can do at home, and I found it really very useful. So how do we measure stomatal length? Well, I've been stripping off the epidermis, put it in glycerin, cover slip on it, and then examining it under oil immersion with a calibrated graticule in the eyepiece. And at times 1,000, each mark on the graticule is 1 micron. So you can actually be pretty accurate with this measurement. Stripping off epidermis is a bit fiddly. If you think stripping wallpaper, sometimes you get lucky and a whole sheet comes off at once. And sometimes you have to scrape around for each little bit, but usually you can get a reasonable strip. So we've checked out our ploidy level by flow cytometry. And I'm hugely grateful to Libor Eckert, who came to the rescue when I locked out of the lab in 2020. And he's been first looking at fresh specimens for me. And since Brexit, we've found that actually silica dried samples are nearly as good. So he's carried on doing that for me. The only way, though, to absolutely know how many chromosomes there are in a nucleus is to count them. So root tips best collected on a sunny morning in early summer and then put into a chemical that arrests mitosis, leaves them in the fridge for 24 hours, and then fix them in 3:1 ethanol to glacial acetic acid, and then use the staining method that Irene Manton used. When I see a likely cell, I photograph it and I print the photograph at A4 size. And then I take that back to the microscope, and by racking up and down, you can follow each chromosome and draw around it. So then the next thing is to number each of those chromosomes you've drawn round. And when you get to 82 for a diploid or 123 for a triploid, well, that's the time to get out the champagne. Doesn't always work. Okay, so now let's get on to some ferns, probably what you've been waiting for. Some ID tips, and these slides are summarized on your handout. So affinis, and this would have been called affinis subspecies affinis. It often has this rather rectangular first lowest basiscopic pinnule, and it's partly joined on to the midrib of the pinna, to the costa, often half adnate. it's quite glossy and robust, but the frond tends to be fairly flat. I'm really grateful to Tony Church for putting me onto the examination of venation. In affinis, the veins are very clear, and the way to look at these is look at the top surface of the frond, hold it up to the light and use your hand lens. So affinis usually has these very thick and rather tall indusia, and after the spores dehiss, they tend to lift and they can split. Okay, so Dryopteris borreri, the texture is one of the first clues. It's much less sort of robust and glossy than affinis is, and it usually has a stalked first lowest basioscopic pinnule. Now, the pinny illustrated here from an example of what I would call Newman's borreri, with the pinnule tips looking as though they've been cut off with scissors. And this is often taken as a clue to borreri, but a warning, it can happen in other affinis taxa. So although you often see it in borreri, it's not in itself diagnostic. The indusia of the covers over the spore cases in borreri are much less thick, they're quite flimsy, and they invert to these shriveled cones and then fall off. So here are two examples of pinnules of Dryopteris borreri. Yeah, I think you'll agree that those veins are much less distinct. The teeth are quite sharp, but the shape of the pinnule is variable. Those first two are fairly common in Ireland, but this one probably isn't. It has quite a stalked first lowest basioscopic pinnule. Actually, I've used the common names that Sellers used. He calls this Welsh male fern, but it's much more common in Scotland. And I think you can see the dark spot is really quite marked. It's a variable thing. It can be quite flat, although the pinnae do tend to turn up in relation to the rachis. And sometimes it's really quite crimped. So I think you can see that the veins, they have some indistinct patches, but they are more marked than in the borreri, and it very rarely has much in the way of teeth. I hope you can see the text behind the behind me, because I can't, but I was letting you read that. The indusia do tend to be tucked under, and then they do all sorts of things after the spores dehiss. So now this is a plant that we found in Waterford, and I think it's very typical of Cambrensis in that it's really untidy, and you can see these sort of very twisty fronds. And a close-up of the same plant, I think this illustrates quite well that the pinnae actually turn at right angles to the plane of the frond, so they look like the steps of a step ladder. So just a recap on the veins, and I must thank Tony Church very much for these photographs. I think Angus Hannah presented this at a Scottish meeting some time back, and it does seem to work quite well. So now on to some more exciting things. This is your Irish male fern, Dryopteris cariensis, and we found this in more or less every place we've been to in Ireland. And I think this is possibly my favourite. I think it's instantly recognisable, and I absolutely disagree with Christopher Fraser-Jenkins that it's a minor variant of affinis. It was thought to be endemic to Ireland, and then Roger Golding found it in Cumbria. And so for my diploid study, I used the Cumbrian plants. And it was significantly different from affinis on 7 of the morphometric traits. I'll let you read that. So some other characters, it tapers markedly to the base, and the lowest pinnae are often tiny. It has these teeth arranged in a fan shape, and it is often quite dark scaled. So that's your fantastic Irish male fern. Now, another one that is in Ireland, but maybe not so common, is this, again, an attractive fern. I don't know why Sell called it greater Scaly Male fern, because it's usually smaller than affinis. It has a stalked lowest basiscopic pinnule. It's crimped. The indusia often go to butterfly wings. And this is not very good photograph of a plant in Galway. A couple of more unpublished Irish specialties. Ken Truran found this plant at the Tork Waterfall and also in Glengarriff. And then Arthur Chater found it in mid-Wales, and they realised that it seemed to be the same taxon. So they were intending to publish it as Atrosquama. Again, it is quite characteristic. And then another star of the show, I think, unpublished again, Ken found it in Glengarriff, hence its provisional name. And this plant is actually at Tork Waterfall. We recognised that there was a different taxon last year, and we called it the crinkly form. It was only when I looked at Ken's plant of Glengarriff this spring that I realised what it was. And then I was able to go back through my virtual herbarium, and yes, the crinkly ones were definitely this taxon. It's dark scaled again. It's got a stalked lowest basiscopic pinnule. So here it is in Waterford. being admired by Anne Trimble and Andrew Malcolm, and there's Megan Morris in the background. It really is a magnificent plant. So here's one in Galway with Roger for scale. The smaller one is Dryopteris cariensis, and the big one is the Glengarriff. It really is just magnificent. And now a very rare fern. So you might recognize this as Arranensis. It was originally found on Arran, and Fraser-Jenkins decided it was a subspecies of Dryopteris cambrensis. But then Anthony Pigott discovered that it's tetraploid, and it has good spores, it makes populations, doesn't seem to be a recent hybrid, and so Cell has published it as Dryopteris pseudocomplexa. I only know of this one location at Drumber Bridge for it, and it is another magnificent plant. And here's Andrew giving a frond some scale. And I mean, it is a bit of a joke, but we do say that one of its features is that it's a bit featureless. The stipe isn't really that robust for a very large plant. Could easily be passed off as maybe a common male fern until you see the clump of it. So just moving on quickly, this one is really contentious. When Stefan Jessen and co-authors published this as Dryopteris lacunosa, it was generally dismissed as just a robust form of borreri. But the distinguishing features are this sort of double tooth. This double toothing seems to occur, and the fjord-shaped gaps, basically funnel-shaped gaps between the teeth. The other thing is it has a long stalk on the pinny, and the lacunosa, that doesn't, the pitted male fern is Cell's common name, but the lacunosa, I think, refers to all the spaces that can be seen when you look at a frond. It is present in Ireland. And then a couple of hybrids. This is the fairly common hybrid between filix-mas and Dryopteris affinis. Abortive spores. So that's what you need to check if you think you've got cross-complex. Now, another unpublished one, although it's in Ken's guide, and as he found it in Ireland, he suggests the name Hibernica for it. This plant is in Galway, near Ballynahinch. It was found to be tetraploid. The Hibernica refers to it being a cross between Dryopteris cariensis and Dryopteris filix-mas. Now, it's very difficult to be absolutely sure which the affinis parent was, but here I've compared the pinny of our presumed Hibernica with those of cariensis, and I think you can see perhaps the similarity with these rather crowded and slightly spoon-shaped pinnules. And then a mystery. So here's Megan and Anne looking at a very odd plant that we found at Drumber Bridge. I have measured the stomata and it seems to be a tetraploid. But its odd thing is that it is so glandular. I think you can see all these tiny little glands over the indusia. I've never seen that before. Christopher thinks it's not significant, but I don't know. And the specimen is in the herbarium. Right, thank you. Sorry it was a bit of a gallop, but I was quite concerned not to overrun.